Okay, so I will just briefly recapitulate that John Kendrew Award has been born out of desire to commemorate the first Director General of EMBL, John Kendrew. It was initially funded through the community of EMBL pensioners, and since 2010, the funding comes from the generous donation from Roland Specker, who is here today, and he will now pass on this sponsorship of the award to his daughter Caroline, who is also here with us. And so please give them a big round of applause. Without them, it's not possible. This award specifically recognizes the former EMBL PhD students and postdocs who have left EMBL for between 2 to 7 years, and it recognizes their achievements during their time in the laboratory, and in particular, how they converted what they learned here into progress in their own independent research. And this year's recipient of the John Kendrew Award will be introduced to us by Nassos Typas, to whom I pass the microphone now.
All right. So thanks, Pavel. It's always hard to take after Pavel and keep up with the jokes. So, all right, I'm Nassos Typas. I'm together with Julia Mohammed. We're heading the Molecular Systems Biology Unit in Heidelberg, and it's my immense pleasure and honor to introduce this year's John Kendrew Award winner, which is Lisa Maier from the University of Tübingen. So Lisa was very briefly, Lisa was an iPod between my lab, Kiran Patil's lab, and Per Bork's lab between 2015 and 2018. I think it's a very great success of how iPod works to do interdisciplinary research on fields that they're not yet established, and this is what I want to pass through today. As you see, I have no slides for two reasons. One, I don't want to go over time, and I want to give the podium to Lisa to make sure she's the star of the thing. The second reason is that there is no reason to show pictures of 10 years ago, because both me and Lisa have not aged a day. We look the same. All right, so So just to give a little bit of history, so Lisa came in 2015 at a time that the microbiome field was booming at EMBL, and the reason for that was actually one single person, and that was Per Bork. So Per Bork was driving the field in Europe, was driving the whole microbiome field based on data-driven research, was mapping who is there, and the diversity of the microbes that we carry in our gut was humongous. The potential of what role they may play for our health was great. But the whole idea is that the only reason we see this diversity is because we don't cultivate things. Otherwise, we would lose the diversity. But that also meant that it was very difficult to go beyond associations. So we can find how things change and associate it with some kind of health status. If we want to go to mechanisms, understand anything, we had to have experimental setups. And that was a big limitation back then in the field. And I think to cut the story short, I think within 4 years, Lisa actually transformed all the experimental research that we do in the human microbiome research. So she was not the first to join those common efforts that we had together with Kiran and Per's lab to set up experimental pipelines for the microbiome, but this was the one that propelled them through. And I think, I would say her legacy still lives at EMBL on approaches that we have here, but not only at EMBL, but all across the field and the microbiome research. So Lisa stayed for 3 years in iPod here. I will not say about the research she did. I think I will let her tell you about the research. I will just say in one line that it basically showed that a lot of the medication that we take have a direct impact on the microbiome, and also the microbiome can change also how medication works for humans. But maybe what I want to say, so Lisa moved on, and she was actually equally successful and even more successful in her own career. So she moved in 2019 with an Emmy Noether to start her lab in the University of Tübingen. By 2022, she got a W3 professor position, an ESC starting grant. She has got numerous awards already. So last year she was featured as the 50 people that basically inspire research from Cell, which I think it's a great thing. And she's also heading a big priority program on functional microbiome research within Germany. So her research is moving a greater trajectory, but I think maybe one thing that is important to say is that she's not only prominent because of her research. I think she takes a very active role in being a role model as a young researcher, as a successful young researcher. She trains the new generation, and she's actually an excellent communicator, and you will see that also during her talk. I think she has been communicate science not only to science audience, but I would say also to general public. She has great podcasts to listen to, interviews, documentaries. So I think she goes beyond the traditional thing of a typical scientist. And I think without due award, without taking any time of Lisa's time, I will just say that Lisa's basically work, if I sum it up in one sentence, calls for a revisit of our drug development pipelines and how we have regulatory policies for drug, which at the moment ignore all the effects that drug have on the microbiome, all the effects that the microbiome has on drugs. And I think that sums up her work. And without further ado, I would be happy that Lisa comes down and we hand her the award and she gives the talk. Slowly, because there's music also.
Okay, so good afternoon, everyone in the room, also to the audience online. It's an extreme honor to be awarded this award. And before anything, I want to thank Nassos for this very kind introduction, the Alumni Association and the selection committee and those who provided support letters, but also Roland Becker for the generous support of the prize over the years. So for today, my task was to prepare a presentation on my life at EMBL, after EMBL, and on our work, so the off-target effects of drugs on the human gut microbiome. And I maybe did the natural thing. I went back to the many pictures that I had from the time at EMBL and let myself fall into the memories. And of course, there were quite a lot of amazing memories from many different lab retreats and lab days over the time, and of course, a lot of things that made me smile. Then I digged a little bit deeper, and I also found my application for the 2014 iPod call. I had written notes on how I would answer the questions, including, where do you see yourself in 10 years? And without going into the details, I would have not guessed in my mildest imagination to be standing here and with everything that is associated. I also formed the program for the interview. And you can imagine, these are postdoc interviews. You have the selection committee, you have the other candidates. It's a public interview. However, there was no one else, no, only the candidates and the selection committee. Unless it was my time to speak, because then the door opened and all the 4 PIs on my grant actually came, sat into the last row and supported me. They didn't ask any questions and then left after my talk. I was floored. This was an immense support by these busy EMBL scientists that I so admired. They took that time off and without me asking, they came to support me. And I think this is really a nice anecdote showing that at least I had from the very first day I came here, immense support that still lasts until today. Very grateful for that. Then, yeah, it worked out. I got the fellowship and I joined EMBL to start working on the gut microbiome. And Nassos nicely introduced it already at that time. It was really a sequencing-driven field. We had good descriptions. We knew about the individual differences in the composition, the taxa that are important, the differences in healthy and diseased individuals, and also that the gut microbiome is important for human health and can contribute to our well-being. It's basically an organ in itself and can signal to distant organs, even the brain. However, cultivation-driven microbiome research was still in its infancies. Many people, in particular the immunologists, believe that you cannot grow these bacteria at all because they are fastidious anaerobic bacteria. And this is why a lot of the work was actually done in mice. So people compared germ-free mice to mice that were colonized. There was not a lot of influence that you had on the colonization status. It was just microbiome there or not, what's the consequences on human health. And on top, we did not know a lot about the biology of these individual microbes. Similar was the case at EMBL. At that time, all over EMBL, we had 2 of these tiny little anaerobic chambers where only 1 person can work in these chambers, and they were sitting on incubators behind doors, not on very prominent places. And there was a brave team in Nassos' lab, in Kiran Patil's lab, but also in Per's lab, that worked really on the fundamentals. So that means putting together the protocols, starting first collections of species that are relevant, but then also what is relevant media. So that's not rocket science, right? That's really the basics. And when I joined, we had a particular question, and that was based again on Per's findings that in patients that suffer from type 2 diabetes, the microbiome composition is different compared to healthy individuals. And then Per and his team, they could show that this is heavily confounded by these patients taking metformin, which is a common anti-diabetic drug. So metformin had a stronger impact on the gut microbiome composition than the disease itself. And that raised the question, how can this be? At that time, it was thought that only antibiotics can have an impact on the gut microbiome. So is this only metformin, or would that be a broader effect? And this is why our aim at the time was to put together a systematic map on the growth inhibition of human drugs or human-targeted drugs on gut bacteria. And the idea was to set up a screen to test 1,200 FDA-approved drugs and their direct inhibitory effect on prevalent and abundant members of the human gut microbiome. On the experimental side, this was together with Michaela Puteanu, and then on the computational side, Michael Kuhn and Georg Zeller. And this took us a while to figure out how to do this in a very efficient way. And at some point, we got actually help by the EMBL workshop. Leo Borger helped us. So at the time, he built a lot of incubators around microscopes, and then we reached out to ask him, would it be possible to have such a stacker-reader combination, and you build an incubator around so that it's everything at 37 degrees. And he took on the task, maybe without knowing that that actually means, so this is the publication version of the figure, but in reality, that means you open here, you crawl in here, you sit in here to figure out what's the best way to have a prototype of such an incubator. At some point we figured out, and we had our systematic map of the drug effects on individual members of the gut microbiome. For the antibiotics, that was the first high-resolution map on different antibiotics from different classes on individual taxa of the human gut microbiome. And you can see that we find different inhibitory spectra by the different antibiotic classes. More surprising at the time was the fact that it's not only metformin that has an impact on the human gut microbiome, but 1 out of 4 drugs that we actually tested. And we found more than 200 compounds that inhibited at least 1 strain from our selection. And they come from different therapeutic classes, in particular the calcium channel blockers and the antipsychotics, and from the antimetabolites. And that result, of course, then raised a lot of additional questions. So what are the molecular targets on gut microbes? Can we use that knowledge to have new antibiotics? How do gut microbes respond and adapt to these drugs? Would that result in resistances and even cross-resistances to human-targeted drugs and antibiotics? What are the consequences for the host? Is this reflected in the side effect pattern? And then are these drug microbiome interactions maybe even required for a drug to work? And if so, can we use this knowledge therapeutically, either to develop new therapeutic strategies or to have more safer or more efficient drugs? I decided to focus on the consequences for the host when I start my little lab at the University of Tübingen. By now, the lab grew into like a bigger lab, really shaping my life after EMBL, quite different, but it's amazing fun to work with this group of people. I think many are joining online and celebrating from the distance. Now, I want to focus on work from Jacobo de la Cuesta Zulaga and Anne Grieshammer. So, what we already knew from the beginning is that the different gut bacteria vary a lot in their drug sensitivity. So these are the individual taxa, and then on the x-axis we have the number of non-antibiotic drugs that inhibit a species, and on the y-axis the number of antibiotics that inhibit a species. You can appreciate that this is a positive correlation, implying that gut bacteria that are more resistant to antibiotics are also more resistant to non-antibiotics. And that implies that you might be exposed to a non-antibiotic drug that then promotes cross-resistances to antibiotics. In the next step, we expanded the screen for a handful of different gastrointestinal pathogens, so the classical Salmonella, Shigella, Yersinia. And I think you can appreciate that they all cluster here, so that means that they are equally sensitive, so are targeted by a similar number of antibacterials. However, they are quite resistant to the human-targeted drugs in comparison to the commensals. And that basically results in a situation where you are exposed to a human-targeted drugs, the commensals are inhibited. That basically frees niches and also nutrients for pathogens to colonize, and then they are resistant to the drugs and can easily bloom in these communities, resulting in an increased colonization and then an increased risk for infection. And again, we can systematically test this across many different communities, many different drugs, and many different pathogens using an easy assay where we have our community and then expose the community to a compound and then first check is there an effect on the growth of the community, on the biomass, and then challenge here with a pathogen, Salmonella in this case, and then have a single luminescent readout that is a proxy on how well the pathogen can grow within these communities. So this is now a little bit of a busy graph, but I will guide you through. So on the x-axis, you can see the drug effect on the community biomass. So below 1 means a reduction in biomass. And then on the y-axis, you can see how well Salmonella can grow in these communities. And then we have drugs here like terfenadine, where we have concentration dependent, a decrease or reduction of the biomass, and that then results in a better growth of Salmonella within these communities. We also have other drugs, like fluoxuridine in this case. It's not a big effect on the biomass. However, the community is changed, and then Salmonella has an easier time to grow in these communities. Of course, that is the next question, whether this would then result in an increased infection risk. And therefore, we turned to an animal model. So we have mice, so germ-free mice, that are then colonized with a human microbiome, and then we treat with terfenadine, so an antihistaminic drug, for 6 days, and then we challenge with Salmonella. That results first in a higher load of Salmonella at day 1, at day 4 post-infection, but then also accelerates the infection with a higher inflammation at day 4 post-infection, which we can quantify with a lot of different inflammatory markers, but then also in histology. So here we really have an example where we have a non-antibiotic drug that disrupts the colonization resistance, so the protection of the gut microbiome against infection. And this is also reflected in cohort studies for a variety of different drugs. We now know the reason, and this is because of the differential sensitivities to these drugs. Meanwhile, at EMBL, I stayed in contact, and it has been amazing to see how the field developed and blossomed at EMBL. So our not very structured, we sometimes meet in the cafeteria meetings, turned out into a very organized microbial ecosystem with additional PIs working on the microbiome field. And also kind of on the equipment side, yeah, there is now this amazing microbial automation and culturomics core facility with all the equipment you dream of if you work in this field. So for me, it's been amazing I feel very privileged to be part of this process, to have kind of helped to get this process started. And I also feel like receiving this award or being awarded with the John Kendrew Award is also kind of a recognition by EMBL's community for this importance of the line of research that we are doing here. We did not, basically stay at EMBL, but then also expanded this line of work, at least to the German community, and it's very proud together with Nassos to coordinate this priority program on illuminating gene functions in the human gut microbiome, where we really want to take this a step further with more microbes involved to push molecular and systems microbiology of gut microbes. Because we strongly believe that in order to, at some point, enable microbiome interventions, we better have to understand the biology, the physiology of these microbes, their genes, and how they are organized into functional units. Yeah, so it's me receiving the award, but in fact, it's actually a whole network behind that are somehow also award recipients, because without them, it would not be possible. That's, of course, my former and current lab members. I couldn't do anything without them. The collaboration partners at EMBL and in the world. Our funding, my home institute at the University of Tübingen, but also my family for their patience and their support. Before I want to close, I want to turn back to my 2014 nervous iPod applicant, and I think I also grew a lot as a person and as a scientist, and EMBL actually played a crucial role in that. I had this amazing support that I didn't ask for, the sense of community, this very tailored career support as well, that of course was given by the community, but for me in particular by my close mentors, so Nassos, thanks a lot, but then also Kiran and Per, and I believe he would be extremely proud if he could be with us today. Thank you.